am showing this product which is made from the medicinal and aromatic plant. Now the crop losses, you see about the virus, phytoplasma, bacteria, fungi and nematode a lot. I am not going to tell you that it is 10%, 60%, sometimes whole crop is going to finish. You will get more than 70-80% loss. It depends on the crop and pathogen. So how to manage these pathogen? The best way is that we have to make the soil healthy. Before coming on the challenges, I would like to discuss about the soil health. When we are talking about the soil health, there are three types. Chemical, it is not working. There is a chemical, physical and biological. When we are talking about the soil health chemical, then we have to go for micro and macronutrient in the soil. When we are going to talk about the physical, then we have to go the water holding capacity, types of soil texture. And when we are talking the biological health of the soil, we have to see how much useful microbes are there. So that is very important. And being a plant pathologist, we are having major challenges. These four challenges. Number one is that to promote the sustainable agriculture and ensure food and nutraceutical security. Not only food, nowadays people are talking about nutraceuticals. So what about nutrition is there? Because there is a lot of problem when there is a nutraceutical scarcity is there. So we have to also need a nutraceutical security to the society. Then to protect the soil and plant health from biotic and abiotic stresses, generally we are talking even about the biotic stresses, we will discuss a lot. To maintain the green and clean environment, this is very important. And when we are using any chemical pesticide, either it is fungicide, nematicide, we have to take the care of the environment health. Why I am taking environment health? Because whatever pesticide we are taking, there is a problem of neurodegenerative diseases. When people are using more pesticide, even farmers are not aware, but you see those area people are having more Alzheimer and Parkinson. Why we are having Alzheimer and Parkinson? Because those farmers, they are not aware how to use these pesticide, how to use these nematicide. When it go in their system, it direct damage their neuron. And when neuron damage is there, the person is having at old age the Alzheimer and Parkinson. So that we have to avoid. That we have to take maximum care, because last week I was in Bangalore in one of the company lecture. Then I asked those people, give me the tox data. What is your toxicological data? Because they were developing one of the product to manage the nematode. I asked them, give me your toxicological data. And that product came in the market. And I am telling you, they were not able to show me the toxicological data. And that product is available in the market. Then I told them, this product is going to damage the neuron of the farmer. They will face the neurodegenerative diseases. Who will be responsible? Then they told, sir, I am taking your mobile number and I am taking your mail ID. I will send you the tox data. More than 10 days over, they have not sent me the tox data. So we have to take more care. When new product is coming in the market, coming in the field, in the farmer field, where is the toxicological data? And toxicological data should be real. It should not be taken through third person. Like I know so many people in Delhi, even they have contacted me when I was working on trichoderma. They contacted me, sir, I can give you the tox data. How you can give the tox data when I am working on trichoderma? I am working on my microbes. How you can give me a tox data? So such type of things is going on. Be plant pathologist. This is our responsibility to take care of these things. Because our major peoples are farmers, society, they should not suffer. This is our responsibility. This is responsibility of the IPS. If some new pesticide, new chemical is coming to manage the phytopathogen, we have to take care of the toxicological data. Then, like European Commission, they have a meeting on 22nd June 2022 and they are planning that they will reduce more than 50% major pesticide in their country. Hope they will reduce and they will ban also. But I don't think in India how we will ban. Even some chemical are banned officially, but they are available in the market. They are available. I am telling you the example of the furadon. When people were taking 2 kg of furadon, it is good for the nematode management. You will not believe, when you take 5 kg, it is not working. You are talking 2 kg, there is 5 kg, it is not working. Because that is not a good one, because it is selling in the market, local market, people are selling in the small city, but that is not active. So such type of examples are available and farmers are not available. And the main challenges is that how to increase the crop production through crop protection. For example, we are having post-harvest losses. Even losses caused by pathogen and pest, if we can decrease it, even we can feed our population another 50 years. Because whatever losses, post-harvest losses and other crop losses are there, even if you maintain, if you manage the pathogen, we can feed our country another 50 years without any problem. The thing is that we are not able to manage them. A lot of post-harvest losses are going on. Population is increasing, no need to tell about this, but how it is possible? It is duty of the phytopathologist, plant pathologist, how to manage the disease. Now the question is that how it looks like in 2030 if damage is continuing. Even though we have to think about beyond 2030 or beyond 2050, even beyond COVID. In one way, the pesticide growth is increasing. You see, 78.16 2021. 2022, they have predicted $85.11 billion. And now they predicted in 2026, $105 billion. So their growth is increasing. We have to increase the biocontrol or microbial management of the pest and pathogen. Otherwise, these pesticides, they are increasing, and what will be the future? We have to think over that. These methods are available. Biological, even consortia is one of the best. During my last 40 years of experiment, I am telling you the consortia is one of the best way how to manage the diseases. One of the example I am telling you that with the chemist, we have isolated one calicarpinine, one of the molecule from Calicarpa microphylla. And when we mix this molecule with the trichoderma and some of the bacillus PGPR, we have got very good plant growth. Even disease suppression was also there. In practical, I have got that consortia is working better than any other way. Even nowadays, people are working on the gene editing, Cas9 technology. They are developing some new plant. They are resistant to pest and pathogen, but sometimes they become susceptible. Even if you see the growth of the biological and chemical, hope after 2050, the biological will grow. It will surpass the chemical pesticide or chemical fungicide or chemical nematicide, as it has been predicted now. And modern biotechnology, they are playing major role for creation of the healthy environment, for soil and plant health. This is a story of the gene technology, how people have developed a resistant variety against the pest and pathogen, especially viruses. For example, transgenic plant disease resistance against microbial, it has been demonstrated in 1986. But when they developed a new papaya against PRV in 1998, its durability was only 15 years. Again, it becomes susceptible. So there we have to think, when we are taking the use of the molecular biology or biotechnology, what is the durability of the resistance? That we have to take care. Then these are the different examples are available, how the biotechnology play major role for the economy of the country or other countries also. I am not going to do it. Now, different omics technology are available. And now people are utilizing these omics technology for developing a plant resistance against pest and pathogen. How we can utilize it against the pathogen and how a plant pathologist can work. I think Dr. Appa Rao is one of the best man in our country who has done a lot on this omics technology. Then when we are talking about the microbiome, it is very important. For example, endosphere. Generally we are, very less people are working on endosphere. But endosphere also play a major role for enhancing the plant secondary metabolite. Why I am talking plant secondary metabolite? Because I am working on medicinal plants and my major role is that to increase the plant secondary metabolite and decrease the plant diseases. So that is my major part. So how this microbiome playing major role we have to think, and we have to increase the useful microbes in the soil. And that will be very useful. Like here, one of the best example you can see when we are using useful microbiome, how the plant yield is increasing. And they are affecting on different parameters. I am not talking one parameter. This figure you can see different parameter is affected by the soil microbiome. So if we are having good microbes, then it is working very well against the diseases. Then if you talk good, bad, ugly, then bad and ugly, how we can avoid it? By using the good microbiome. So here we have to take the indoor rhizosphere and rhizospheric microbes. Both we will take and then we can increase the yield of the crop and develop resistance against the pest and pathogen. Here one of the best example I am showing that how the plant develop a immunity, either it is ISR, either it is SAR, by using the different microbes in the plant. Nanotechnology, now growing area of the nanotechnology. People are using nano fertilizer, nano pesticide, nano fungicide, nano nematicide, and it is working. But we have to think these nano, they are not going to enter in our food chain. That we have to take care, because if nano is coming in our food chain, then it is going to have a great impact on our health. So that we have to take care when we are working on the different type of the nanoparticle. And even ketosane, Dr. Podeli is working, Apparao is working on ketosane. He know better how people are using this nano ketosane, and they are mixing these ketosane with the different molecules, plant molecules, and they are making that anti-biofilm activity, because whatever the pathogenic organism are there, they are making a biofilm. Biofilm means then our useful microbes cannot enter. So until you are not going to break the biofilm, whatever technology, whatever things you are developing, whatever microbes you are developing, it will not work. So that is major part is that how to break the biofilm. Then our molecule, our things will work. So these are playing a major role, and that we have to take care. And then nanopesticide, already I have discussed, nanofertilizer, no need to discuss here. And I want to show you some of my own work, because what I did, what my group has done. Here one of the example I am telling you that we have taken trichoderma and some of the microbes. And what we did, we have developed a lignification against the nematode. So when we are developing lignification in the root, nematode cannot enter inside the root. So we are restricting nematode in the soil, they cannot enter in the root. So here we manage the nematode problem by using the microbes, by increasing the lignification and increasing the plant secondary metabolite, bidafrinate. That is the major part of our research. Then here another point is that we are working on aromatic plant. Aromatic plant yield, only oil can increase when we are increasing the number of the trichomes, because trichomes are main part, they make the oil gland. So here when we have used the microbes, they increases the number of the trichomes, and when trichomes are increasing, then we are getting very good maximum oil glands. So oil yield is increasing. So here we were targeted in aromatic crops how the microbes can increase the number of trichome in the plant leaf. That is our major part, and this work we have done and published it. Then another is Bacopa monday. This is very important plant for the memory tonic, and bacoside is the major active molecule, and we have used trichoderma and Bacillus subtilis, and we have increased the yield of the bacoside, because our target is that to increase the plant secondary metabolite, not only increase the plant yield. If you are increasing the plant yield and alkali yield is zero, there is no value. So ultimately we have to go to target molecule or target alkali. When we are increasing the yield of target alkali, then only it is useful. Otherwise, there is no useful if we are making more herbage or something like that. And then we have used some of the Chitinophyllus and Streptomyces against the root knot nematode. You can see here the lower figure. In control and nematode is there, you see whole root is damaged. But when we have used microbes, we got the less damage in the root system. So how we use the microbes to manage the nematode problem in the plant. And not only I am showing you the practical, we have developed a technology also. And that technology has gone to the farmer field. It is not only I am publishing an impact factor journal and showing you. This technology has gone to the farmer field. So that is the beauty of our science. Because in CSIR, if your technology is not going to farmer field, it is very difficult for you to get a promotion. For scientists now it becomes very mandatory. So we have to work for the society, for the farmer, if we want to grow our career. That is very important now. And some of the picture I am showing how these different microbes manage the reactive oxygen species. You can see in the leaf, then in the root, you can see how we are increasing this thing, how we are managing the nematode problem. And then we have taken the picomolar solution of some of the molecule and we have increased the microbial population in the root system. Means in the root, we have got more colonization when we have taken the picomolar solution of the butene. That paper we published in Platinum. And then another work we did here, hexavalent chromium. Because in the nearby Lucknow there is Unnao and Kanpur district. Lot of the plants are affected by the hexavalent chromium. And these hexavalent chromium not only affecting the plant, they are carcinogenic also. So what happened, we have isolated some of the Bacillus megatherium species, later on we identified it, and that make hexavalent chromium to trivalent chromium. Because trivalent chromium is non-carcinogenic. So that microbes not only remediating the hexavalent chromium to trivalent chromium, it is also increasing the IL yield and decreasing the nematode population. So these are the different parameters I observed. So I am not going in deep because time is very less. And another thing, this is our technology, how we transfer this trichoderma technology, what patent we are having for patent. That technology has gone to farmer field, and if any technology is going to increase the yield of the farmer crops, then it is good. For example, if I am working, I am becoming a trichoderma man. If farmer is affected, farmer is getting yield increase, getting more money, more revenue, then it is useful. Otherwise, there is no use of any technology, just publishing a paper. So here, how we use the sucker, then minister and governor, they all visited our whatever product we develop in our lab. But the most important thing that I am happy because I am seeing happiness on the face of the kids. And at least the farmers who have adopted our technology, they are having very good revenues and they are making their pakka houses and they are making even from ox, they have purchased some tractor and other things. So now they are working very well when they adopted our technology. And whatever I did, because I have not shown my all work, only few slides I have shown you, this credit goes to my student, not to me. I am not responsible. Whatever I did, whatever I published, these all credit goes to my student because they are the major pillar of my science. What science I did in 40 years, these all credit goes to my student. So thank you very much. And this is my herbal garden, Manav. We have made a garden that every medicinal plant should be in the different part of the body. So at least the student can identify and see the use of the plant.
All of us enjoyed the presidential address delivered by Dr. Rakesh Pandey. He has brought in his rich experience of working with medicinal plants and development of different products. Sir, as a customary, I believe presidential address will not be open for questions. So on behalf of the organizers, on behalf of our Indian Phytopathological Society, I thank him. He has reduced his lecture time and saved a little bit of time for the session and for the forthcoming sessions also. Thank you so much, Professor Rakesh Pandey. We now have another important plenary speaker, none other than Professor K.V. Subbarao. Professor Subbarao is a son of the soil, graduated from this university, obtained his master's in 78, and then briefly he worked with ICRISAT for about 5-6 years before he went on to do his PhD at Louisiana State University in the United States, and which he graduated in 1989, for which he got a very prestigious award, the Edgerton Award, CW Edgerton Award for Outstanding Performance as a graduate student at Louisiana State University in 1988. And to mention very little about Professor Subbarao, he is currently a professor at University of California, Davis, and he was also associate professor there. And most of us would have seen him on the prestigious journal that many of us would look at, the Phytopathology. He has been associated with the journal for more than a decade in different capacities, including editor-in-chief. That's a prestigious honor that such kind of and he is an elected fellow of the American Phytopathological Society. With all these distinctions, he has agreed to be with us to deliver his plenary talk, and it is my pleasant duty to invite him to share his experience on comparative genomics, particularly on Verticillium dahliae, where there is very little work on Verticillium dahliae, and he is going to speak to us on the comparative genomics of defoliating and non-defoliating strains of Verticillium, and how the horizontal gene transfer from another wilt-causing organism would have been responsible for all that. And with this little of introduction, I invite Professor K.V. Subbarao for his plenary talk. Professor Subbarao.
Dr. Padile, for that kind introduction. My heart swells with pride today because I learned the ABCs of plant pathology right in this very campus. And it also infused in me the power of observation, whether it is under the microscope or in a research field or the general changes in the society that directly affect plant diseases, that those experiences went on to shape my career, and I'm very happy to be with you and present this talk. I know you're all hungry. We are already 2 hours late. I won't take too long, but I want to just give you a glimpse of what is possible. These are exciting times to be in science, and especially in plant biology, because of the technological changes that are available now. The kind of problems that you can solve are simply limitless. And I'll give you one example of such a problem that had existed for over 70 years, and using genomics, we were able to resolve those issues. And as you may have heard my talks in previous APS meetings, I have had a long-standing interest in wilt-causing pathogens, especially Verticillium, wilt caused by Verticillium dahliae. Pointer work color. What's happening? All right. So Verticillium wilt is caused by it's a small genus, Verticillium. It has 10 different species, and Verticillium dahliae is the most important one. So Verticillium dahliae is the most important pathogen. It's a strictly soil-borne pathogen. It produces long-standing survival structures called microsclerotia that remain in the soil viable for up to 10 years. And once it is infested, you have extremely long-term problem in your fields. It causes the vascular wilt, it invades the xylem tissue, causing the wilt disease. And it also has a very broad host range, conservatively estimated. It infects up to 250 host species, many of them including economically important crops. And it is an entirely asexual pathogen. No one has observed sexual cycle either in the laboratory or in the field. So it is a clonal pathogen. On the right-hand side, you see some of the crops that are affected by the disease. And the plants nearing harvest will simply collapse with the wilt because the xylem is completely plugged up. So wilt diseases in general have three different phases. The first phase is at the root zone. That is the initial combat zone, and in the xylem tissue, that's in the middle, and the ultimate or final victory of the pathogen over the plant happens in the leaves by way of this one. And different components of the pathogen actually played different roles at each of these phases that are shown in the different colors on the left-hand side. And the entire array of enzymes and toxins that are produced by the pathogen, we call them secretome. The different components of the secretome play different roles in each of these phases. So we published a review just last year in Biological Reviews, and if more of you is interested in studying the details of this one, you can refer to that one. And I told you it is a pathogen of upwards of 250 crops, and in all of those crops, the normal symptom that you observe is wilting, except in three crops that are represented here, cotton, okra, and also olives. The wilting is combined with defoliation, premature defoliation of the plants. Certain strains of this pathogen cause complete defoliation, and on the left-hand side you see a branch of an olive tree, 2-year-old olive tree that is already deceased. By the end of the second year, it is completely denuded, and by the end of the third year, the plant is completely dead. If you cut open the plant, then you see the characteristic vascular discoloration. And in the cotton, exactly the same thing happens. Just when the plant begins to flower, it sheds all of the leaves when it is infected by certain strains of Verticillium dahliae. So defoliation has many causes. We have learned that it is a natural process of growth and development that is mediated by these two phytohormones, abscisic acid and ethylene. And certain phytotoxic compounds also cause defoliation. In fact, the two compounds that are within the parentheses there are actively used on cotton to defoliate prior to machine harvest because they don't want the lint to have too much of a foliar debris. So deliberately the cotton crop is defoliated using these two. And we also know there are plenty of abiotic stresses that cause defoliation. And defoliation can also be caused by both pathogens and pests. So the phenomenon of defoliation was first discovered in California, in fact. There was a USDA scientist at UC Davis that discovered this phenomenon in cotton, and he characterized the strains that cause defoliation and non-defoliation and called them as such, defoliating and non-defoliating. And since then, there have been over 100 papers on this very topic without completely addressing why defoliation happens. And many groups have tried and failed, and it is only, it probably waited for us to resolve this issue, and I'll share that story with you. So previously, we had used a genomic, comparative genomics approach to study the differences between race 1 and race 2 of Verticillium dahliae, where we obtained the complete genome of these two and compared the two genomes. And in race 1 isolates, about that band that you see, the 50 kb region, was only present in the race 1 strains, and it was completely absent in the race 2 strains. And we went on to characterize a race 1 effector from this region that was published in PNAS in 2011. And we used a similar approach in this study also, wherein we selected a very virulent strain, VD991, that is defoliating from cotton, that is not only virulent on cotton, but is also moderately virulent on tomato, but it is completely avirulent on lettuce. And similarly, we also selected a strain from lettuce that is non-defoliating, and also the strain from tomato that was also non-defoliating. And as you can see here, only the defoliating strain causes defoliation, and none of the others defoliate the cotton plants. And only cotton is infected by all three strains, and lettuce is not is infected only by the by the lettuce strain. So we obtained the complete genome. These are 8 chromosomes. There are 8 chromosomes in Verticillium dahliae, and on chromosome 3 and 4, there are these specific regions that are called lineage-specific regions that are very rich in transposons that confer some of the specific characteristics to Verticillium dahliae. So when we compared those two, in GSLR2 that is shown in the red box, that was completely different in the defoliation strain than the other 2 strains. And we took that area to study further. So we obtained the complete genome to ensure that it was not specific to VD991, the strain that we selected. We compared a whole population of the defoliating strain, and in all of those strains, you see the presence of that 109 kb region that is absent in the non-defoliating strain. And we were curious to find out where this region came from. How did Verticillium dahliae defoliating strain acquire this region from? So we did a phylogenetic analysis comparing it to several species of Fusarium, and you notice that small box that is shown in green, that was complete, that had a perfect match with Fusarium oxysporum vasinfectum, which is also a wilt-causing pathogen on cotton. So there was actually a horizontal gene transfer from this, and it can easily happen because they both share the same niche in the soil as well as in the plant xylem. So we knew that it acquired, once it acquired, there were some deletions and additions that conferred the cotton strain of Verticillium dahliae this specific defoliation ability. So we then went on to characterize functionally how this region actually causes defoliation. And as you notice here, when you delete this GSLR2 that is in the cotton strain, the defoliating strain becomes the non-defoliating strain on cotton, but it has no effect on the other 2 hosts that we included in this study. So when you transfer that GSLR2 to either the tomato strain or the lettuce strain, it actually increases the virulence of those 2 strains on cotton, but it doesn't alter the virulence on either their own respective hosts. So this region we knew is the one that causes defoliation. So the next question is, how does it cause defoliation? So we then went on to, this is once again on lettuce and tomato. There was absolutely no effect of transferring this on the virulence of these 2 strains on their respective hosts. So we knew that horizontal gene transfer of this LSR region is what conferred this defoliation ability to Verticillium dahliae from cotton, and it is probably one of the mechanisms by which asexual pathogens develop virulence mechanisms also. So there are 7 different genes within this GSLR2 region, so named VDDF1 through 7, and we individually knocked out all of these, and then we assayed their virulence. As you can see here, from defoliation strains of gene 1, 2, 3, and 4 did not have a phenotype. In other words, they still remained as a defoliation strain, even though they lacked the corresponding gene. However, when you delete genes 5 and 6 in this region, then the defoliation strain becomes the non-defoliation strain. So it is these two genes that are very, very important in conferring the defoliation. So you notice that gene 7 also did not have a phenotype here, but it plays a very significant role indirectly, and I'll come to that in a couple of slides. So the question then is, since it acquired this region from Fusarium, whether the GSLR2 from Fusarium, or these two genes, 5 and 6 from Fusarium, are able to cause defoliation. And as I told you before, it does not cause defoliation simply because these two genes have an ineffective expression. Both need to be expressed at the same level at the same time, whereas in Fusarium, it does not happen. And because of that one, defoliation does not occur with Fusarium oxysporum vasinfectum. So then we wanted to compare the results on cotton with also olives, just to ensure that what we are observing on cotton is not limited to cotton. So we repeated those studies on olives with identical results. So what is special? How do these two genes cause defoliation? What do they encode? In fact, both DF5 and 6 are in the PKS cluster, so they encode that compound N-acyl ethanolamine, NAE, commonly called. And DF7, I told you, it did not have a phenotype. What it does is it encodes an enzyme that feeds more of NAE into the system itself. So that's how it plays a very significant role. So there are a number of different NAE types, and this is the flow chart for how the enzyme that VDDF7 encodes produces more NAE to feed into the system itself. There are different types of NAE, and they have various functions in the plant systems, and we wanted to find out which of these NAEs are produced by Verticillium dahliae and which actually cause the defoliation. So Verticillium dahliae produces 3 different NAE types: NAE16, NAE14, and NAE12. And on the right-hand side, you see the amount of NAE16 that is produced by both the defoliating strain and non-defoliating strain. The variation within the strains of individual group were much larger than the differences between the groups themselves. So obviously, NAE16 was not the candidate that explains the difference in the two strains types. And NAE14 is produced more by the non-defoliating strain, and obviously this was not the candidate that causes defoliation either. So then we settled on NAE12 and did functional analysis once again, and as you can see here in the in the strains where genes 5 and 6 of this cluster are deleted, their ability to produce this NAE is severely restricted, whereas the wild type strain produces copious amounts of NAE. And if you complement those two genes in a strain, then it restores the ability of that strain to produce NAE12. So then if you put these two genes in a non-defoliating strain, whether that is able to produce this NAE12. So that is the last strain that says VDG78. That is a non-defoliating strain into which we added these two genes, 5 and 6, that is able to produce NAE12 once again. So the question then is, if you know that NAE is the chemical that causes defoliation, whether you can reproduce the defoliation symptom in the absence of the pathogen, or if you inoculate the plant with a non-defoliating strain, if you supply NAE12, does it also defoliate? That is the experiment that we did here in the bottom that you see, where we inoculated the plant with a non-defoliating strain, and then 1 week later we supplied NAE12 through soil, and it immediately was able to defoliate the plants. And if you actually simply supply NAE12 to the cotton plant, that also causes defoliation. but it takes much longer. In the presence of the pathogen, the defoliation happens much quickly, but NAE12 alone is also able to cause defoliation, but the whole symptom onset is delayed. And we hesitated to do this experiment, and we submitted the manuscript to New Phytologist as it is, but the reviewers and the editors insisted on us doing this experiment. The reason we had not done that experiment is because this NAE12 is very, very expensive. This figure that you're seeing here in the bottom, that alone costed us $35,000, just that one experiment. So we had to bite the bullet and do that experiment to produce those results to convince the reviewers, and finally we were able to do that one. So there are a number of functions of NAE, as I told you in here, and we wanted to further study how NAE causes defoliation of the cotton plants. The way it does is it induces this overexpression of fatty acid amide hydrolase genes in cotton, and this overexpression affects the hormonal balance, and because salicylic acid and jasmanic acid production are completely rendered useless, then it makes the cotton plants more sensitive to ABA and also pathogen infection. And this is how the plants are able to defoliate, the strains are able to defoliate. And here in this figure, I'm just showing you the number of fatty acid amide hydrolase genes that are overexpressed in a defoliating strain as opposed to the non-defoliating strain. So that is to conclude. I just want to give you a story. In fact, in the University of California system, for all graduating PhD students, there is a competition in each of the campuses. So their entire PhD work has to be summarized within 3 minutes. And then if the winner will go on to get a presidential award, in fact. It's a very prestigious award. And I don't need actually 3 minutes. I have summarized all the studies in this one sentence that you see on the right-hand side. So I would like to thank all of my colleagues and also the funding agencies that supported this study. And before answering your questions, I want to leave you with this quote of a Zen master, Suzuki. And the challenge is to maintain that beginner's mind. Beginner's mind has a lot of possibilities. And if you're able to maintain that beginner's mind throughout your career, success cannot be too far behind. So with that, I'd like to thank you for this opportunity.
Now we can see publications for PNAS, new phytologists, and physiologists. All of them are talking about the quality of the data and the kind of conclusions that they have come up with this. I would now like to keep this talk open for a few interesting questions if any one of you have, because let's not worry about lunch. Lunch can always be taken. It is good to have an interaction with a scientist like Professor Subbarao here, and not to bother too much about lunch there. Professor Dubey first. Up mic, please.
Exhaustive and complete story you have given. Two points in my mind. Whether what are the evolutionary process for the verticillium, whether the initially defoliating species came or non-defoliating species?
It is the non-defoliating strain that is prevalent, because in nature, these are the only 3 hosts on which non-defoliating strains exist.
That means the virulence has been increased when it's become the defoliating.
Exactly. Yeah.
So what was the time spent required to differentiate these two?
Yeah.
Because you have not so many physiological mechanisms how the defoliation has been, the chemical factors which are required for the defoliation, but what happens structurally over there, what kind of the signal is going, why they are detaching from the, what kind of the virulence pattern has been increasing, whether time is reducing or increasing.
Right.
That's
So we didn't put a molecular clock on this phylogenetic tree because that was not the focus of the study. We didn't want to know. We knew that defoliation strains have been there for a long time. even though they were only discovered in the 1960s, they must have been present much earlier than that one. So who knows when it happened, but what do they do once this virulence mechanism is acquired is what we discovered.
Okay, thank you, Professor Subrah. Professor Chahal has the point.
Well, excellent work, and I must congratulate you. It's a very beautiful job you have done. This defoliation is really a problem in many crops, not in this particular host pathogen system. Verticillium is one, but other pathogens are also having a similar effect, particularly on field crops. The question coming to mind was that we are facing in agriculture a lot of problem in cotton, a lot of problem in other crops. Say, for example, last year what happened, that during this grain drilling stage in wheat, there was a heat wave. Climatic conditions are variable and very indifferent, causing a lot of damage to this crop production. Last year it happened in India that production was gone so down that it was less than even the previous year's total grain production. So it has very wide implications over my, it was coming when I thinking that not only this host pathogen system but in others too. What is your take about this when we come to these climatic variations and how this can work there and can affect it?
Yeah, I mean, defoliation, as you know, it's a natural process. It's a natural process of aging. And it only becomes a problem if there is premature defoliation. So that's what affects crop productivity. And you mentioned drought stress. Obviously, that is one of the factors that I indicated that also causes defoliation. So how do you resolve that one? There is no way to overturn a drought situation immediately. So we have to breed for drought-tolerant crops, and that many groups throughout the world are doing it actually. And in fact, even when I was at ICRISAT, there was a large-scale experimentation on drought tolerance in pearl millet as well as in sorghum. So that would be the way to go, I would think.
Yeah, Ashwini Parikh is at Punjab, sir, in Chandigarh. Ashwini Parikh is engaged in this drought-tolerant development of drought-tolerant varieties. Yes.
So Subha, it's very nice. You explained very nicely and through experimental procedures. But it is very interesting to know that why this virulent one defoliate. Because if we logically think that the pathogens would like to survive longer time in the plant, infected plant.
Yeah.
So, but the virulent one is going to defoliate very prematurely. So there may be other, can say, strategy plant pathogen is going to have. Because I think the reproductive stage, if the plant is not there, so reproductive stage will be much more faster. So can you correlate these things? Because I am just curiosity in my mind that how a virulent one is going to defoliate it much faster way compared to the.
There is a very easy answer to that one, I will tell you. So because only 3 crops defoliate, and this pathogen has to survive on those 3 hosts only. So when it defoliates, it is able to produce those microsclerotia on those defoliated leaves. So it is able to reproduce there faster, and that is its survival mechanism.
That's all. Thank you.
Any beginner's minds?
Yeah, exactly.
Myself, Dr. Bilal. So you said about evolutionary things. Have you, sir, done about selective sweeps? Have you seen selective sweeps have much role to play in evolutionary of this effector genes?
In certain pathogens, exactly, selective sweeps do happen. But in this case, I don't think that is the process.
It was comparative genomics might have given you some selective sweep ideas that if this pathogen, rather than having horizontal gene transfer, might have a selective sweeps to evolve those type of PAMs?
No, I don't think so, because we found a perfect match with the sequences of the Fusarium oxysperm infectum pathogen. So selective sweeps is a mechanism in several other pathogens, but not in Verticillium dalium. Yeah.
Any other questions? Yes, Robin?
So thank you, Dr. Subbarao. So your gracious presence and excellent talk improved the quality of this symposium and also annual meetup in the Pathology Society. Very simple question I want to know. After seeing very nice story, scientific story, what we have witnessed just now. So verticillium, by virtue, it is a vascular pathogen. And you have shown differentiated between defoliated suppose isolate as well as non-defoliated isolate. Now, did you compare how fast defoliating isolate can multiply in the vascular bundle as compared to non-defoliating this isolate?
We didn't study that specifically, but I can only speculate. I think it ramifies, once it enters into the xylem, it ramifies much faster than the non-defoliating stream. And that's how it is able to cause that defoliation. Because the more it reproduces, the more NAE it is able to produce and infuse into cotton. And once it infuses NAE into the cotton system, that results in the overexpression of that fatty acid amide hydrolase genes. And that makes it sensitive to ABA action. Yeah.
Thank you.
Okay. Yes, Hamida.
My question is from the NEA. You said the NEA was not sufficient to defoliate. So is it a quorum sensing molecule because the pathogen was also is also necessary. So in verticillium, can we call it as NEA is acting as the sensing to induce the pathogenicity of defoliation?
No, it does, as I just told you, it results in the overexpression of the cotton genes. It makes it more sensitive. So that is the function of NAE.
So it's not with the lipid signaling or because fatty acid hydrolase is also related here?
It is possible. It is certainly possible. But the direct conclusion that I can make is this one. So, yeah.
Okay, I think there are no more questions, and I would like to thank both Professor Rakesh Pandey and Professor KSOU for the wonderful talks on behalf of the Indian Pathological Society and on our own behalf. And I would like to request my co-chair, Professor Sridhar, to summarize and close the session.
So we had two very interesting lectures by the presidential lectures by Professor Pandey and also Professor Subbarao has given a very exciting information about the genomics, especially with regard to the disease. And I have one question. Is it possible to prevent the horizontal gene transfer?
No, I don't think so.
Any possibilities?
No. The horizontal gene transfer has been happening for millions of years.
Yeah,
from
it's a evolutionary process.
Bacterial systems to pathogens or plants. And in fact, I'll tell you, the race1 effector that I told you in Verticillium dahliae, it was originally a plant protein. It came from the castor plant. So it ended up in Verticillium dahliae. So how can you prevent it? No.
Okay. So thank you very much, sir. I thank both the speakers profusely for their excellent presentation, and also it has created a lot of interest to discuss with them, and I hopefully we can discuss the rest of the things during the session. Thank you.
Thank you very much. And in fact, when Robin sent the email to all the membership, I must have been the first one to reply to him that I'm attending this meeting. I was so proud. Thank you so much.
Genomics resolves historic puzzles in plant pathology. And now we'll honor Dr. Krishna V. Subbarao. And I request Dr. S.C. Dubey, ADG, Plant Protection and Biosafety, ICR, to please come on the stage to honor Krishna V. Subbarao, sir. And also I request chair and co-chair to join in. And I request Dr. S. Chandranay, organizing secretary, and the president of IPS and the secretary, IPS. Please, sir, please. And I request Thank you, sir. Thank you very much. It's our pleasure having you in Mysore. And I request Dr. President IPS. I request Dr. Chahal, former President IPS and former VC, MPUAT Udaipur, sir, yeah, to honor Dr. Rakesh Pandey. Sir, please. Thank you, sir. Thank you very much.
So that one is very fast, very fast.
And this co-chair as Professor KSOU, Sridhar, Mangalore University, Mangalore. And I request our organizing secretary to present a token of memento to Dr. Harleen Kaur, Punjab Agriculture University, Ludhiana, Punjab. And to Dr. D. Parmesh, AICRIP. And today's session is chaired by Dr. R. Vishwanathan, ICR Indian Institute of Sugarcane Research, Raebareli Road, Lucknow, and he is a scientist and ICR Indian Institute of Sugarcane Research and head division of crop protection, ICR SBI, and also he served as a principal investigator AICRP on sugarcane and a principal scientist head and division of crop protection, ICR Sugarcane Breeding Institute, Coimbatore, Tamil Nadu. And he has been awarded with many prestigious awards like MS Rugby Memorial Award in 2020 and Indian Phytopathological Society, New Delhi, and Shri Syed Ahmad Khan Memorial Award, Aligarh Muslim University Centenary Award in 2021, and the Sugarcane Technologies Association of India Noel Day Gold Medals in 2002 and 2022 consecutively, and Sugarcane Technologies Association of India Silver Medal in 2001, and so on to his credits. I welcome you, sir. I welcome you to the session. Dr. R. Vishwanathan. And this session is co-chaired by Dr. M. Krishna Reddy, ICAR, IIHR Bengaluru, Karnataka, and is presently working as a principal scientist head division of crop protection, ICR, Indian Institute of Horticulture Research, Bengaluru, and is area of expertise in the field of plant pathology, plant virology, molecular plant pathology and biotechnology, host pathogen interaction, molecular diagnostics, and virus resistance identification and development of resistance genotype, epidemiology, and integrated virus disease management. And he has also been awarded with many prestigious award recognitions and so on. I welcome you to the session, sir. And Today's session rapporteurs are Dr. Prakash TL, ICAR, IARI, Indore, Madhya Pradesh, and Dr. Amrita Das, Division of Plant Pathology, ICR, IARI, New Delhi. Welcome you, ma'am. Welcome you, sir. We'll start with the keynote lecture, and I request our chair to introduce Dr. Apparao Kodle, today's keynote speaker. Thank you, sir.
Good afternoon. Plant pathologist of the country and globally, we can say that Apparao Kodle has contributed immensely on post-pathogen interaction. So we are happy to support this audience. Do not require so much introduction about Dr. Apparao Kodle. So he is a man of such contribution and served as Vice Chancellor of the prestigious and then assumed had several positions to his credit, and also you must have can see that he is one of the few scientists in the country who have been decorated with fellowships from all the major academies in the country. So that shows his contribution in the field of post-pathogen interaction, and almost I know his work for the last 25 years plus. So he has been consistently working on that, and many of his students are in different laboratories elsewhere in the other countries. That also speaks about his contribution of training the young generation on this new area of post-pathogen interaction. And he, as I was reported that he is apart from fellowships from academies, he has been adjunct professor in Charusat, Gujarat, adjunct chair professor in biotechnology, Asia University, Taichung, Taiwan, member of Indian Research Group, International Research and Training Group on Molecular and Cellular Glycosciences, supported by DFG in Germany. He is a visiting scientist in Department of Plant Biochemistry and Biotechnology, University of Münster, Germany. And he was also visiting scientist, Institute of Molecular Biology, Academia Sinica, Taipei, Taiwan. Postdoctoral fellow, National Science Council, Taiwan, at the Institute of Botany, Academia Sinica, Taipei, and many other places where he worked. And apart from that, the various, I am just reading a few honors. Apart from fellowship, he was member, Executive Council, National Academy of Agricultural Sciences, New Delhi, EC member, Consortium of Academic and Research Ethics Committee, University Grant Commission, Adjunct Chair Professor, Department of Biotechnology, Asia University, participant Fulbright US-India Education Foundation, sponsored by Fulbright Academic Administrator Seminar, November 2019, KC Mehta Memorial Biennial Award, National Academy of Agricultural Sciences, New Delhi, JC Bose Fellowship, Department of Science and Technology, President Association of Microbiologists of India, Honoris Causa, that is a DSc from Ram Manohar Lohia Harvard University, Tata Innovation Fellowship from Department of Biotechnology, and he has more than 33 years teaching experience and completed more than 2 dozen external funded projects, and even now he is continuing with many funded projects. I invite Dr. Apparao Kodle to deliver his the first keynote lecture.
Thank you, Chair Dr. Viswanathan, and Co-Chair Dr. Krishna Reddy, distinguished professors and participants. I was thinking that I will do it in about 25 minutes. Now I'll try my best to do it in 20 minutes since Dr. Krishna Reddy indicated about 20 minutes time. There is something, the topic that I have chosen to discuss with you is to introduce some concepts which are relevant today, where we need to really think about how to understand the plant pathogen interactions to move forward and that's one reason why it may take a little longer if it is only the second part of the lecture would be only about our work that we have done. So since I'm combining the both, I thought it may take about 25 minutes. Let me see if I can do it a little faster. So basically my interest is in today's topic is chitosans. Chitosans, we have been working for quite some time, and in that we have done more significant work during 2005 to 2022, which has been the last 15-17 years, and I will share some of those things with you. This is a slide which is quite familiar to most of the plant pathologists sitting here. The two types of immunity that we often see in the plants, that is the PTI and the effector triggered immunity, and in between you also see effector triggered susceptibility. These are the two major immune responses that have been characterized so far, with so many details available for both PTI and also for ETI. The essential part of it is there are pattern recognition receptors and there are pathogen associated molecular patterns. If these two things we are take home message from this slide is that, and this is one great review that has appeared, that appeared in 2019, the Tansley review, where the responses, because this pointer I will not be able to use, the responses that you find in the ETI and PTI, you can see they are very simplistic presentation of how the plant responds in PTI and ETI, and also with the viruses and all that is shown in this particular slide. And I will skip this slide. And this is a slide which I got impressed a couple of days ago when I was preparing for this particular talk, where there was a paper in Nature Plants that appeared in October 22, where a comparative genomics of 350 plant genomes has been done, and where the evolution of these the pattern recognition receptors, particularly the extracellular ones and the intracellular ones, has been studied in detail, and a lot of information has been brought about by using this kind of a comparative genomics approach. Some glimpse of it we have seen from pathogen side, which was presented to us by Professor Subrao in this morning. And this is the source for chitin. Primarily the source that is used in the market for the chitin is the shrimp shells, and when you prepare the chitin you get a pure form of chitin in this form. But this is an insoluble form which is crystalline and it is insoluble. And to make it soluble, most of the time you will have to do a deacetylation process. When you deacetylate the chitin, you get chitosan. Of course, the degree of deacetylation varies, and then next things I will introduce. And when you deacetylate, there is a scope for solubilization of chitin, and it is the soluble forms of these molecules which can be recognized by biological systems. And as chitosans, as functional molecules, we are talking about the chitosans. When I go to the next slides, you will realize it is not simple chitosans. The degree of acetylation on the polymer or the oligomer, degree of polymerization of the oligomer, and pattern of acetylation of oligomer, all three of them are going to be crucial. And it is